Okay, great. So thanks everyone for coming to MIA seminar today. I guess now after daylight saving, so it should feel a little bit more awake. But yeah, so today we're very lucky to have Jingyi Jessica Li here and postdoc Pan recently moved to the Fred Hutch Cancer Institute. And today they're going to be talking about some exciting new statistical work they've been doing. And so I think Jessica is going to lead off on permutation enhances the rigor in genomics data analysis. Thanks so much for coming.
Thank you, Taylor, for the invitation. And it's our great pleasure to talk about our research at the MIA seminar. And so my talk will be the primer, and then Pan will give a specific talk on her research. So my primer hopefully can open the door for her talk. And I'll introduce some basics and our thinking in particular in this research. So the theme of the talk today is permutation, but later I will go broader to talk about how we think using something we call as the synthetic node data can help with our data analysis. I think we hope this to be broad enough to be relevant to your research. So I'll begin by this interesting figure, which was actually something we found on social media after the Nobel Prize in physics was announced last year. And Pan actually helped me do the translation because it was originally in Chinese. But I found this very interesting because the question is, what is physics? And it can be characterized in two dimensions, the objects we study and the methods we use. So you can see that for either dimension, we can go from being very fundamentalism to very liberal. So it means that we can be very restrictive on what we study, the objects, whether there's something very general, or the methods we use like laws, or we can be very liberal. And the neural network is actually on the liberal side for both dimensions. We don't use physical laws, and the object we study is not something we see as concrete in real life. So this kind of characterization made me wonder, how about genomics? So for many reasons, genomics can be seen as a liberal discipline, as many of you may be aware of because of your research, you can judge. Because it's very interdisciplinary, you may be trained in a very wide range of disciplines, and it's very data-driven. So we start from data, and there can be all sorts of hypotheses we can ask. And there's a rapid revolution of methods. Just using the single-cell RNA-seq as one example, you can see this rapid evolution of methods, thousands of methods. And as a result, you can have flexible analytical approaches. How do you design the pipeline for your data analysis? There can be many options. Since my research is at the intersection of statistics and genomics, so I was a statistics professor at UCLA for 12 years. So if I think about statistics, it's very different from genomics because people there really talks about rigor. How can we have a better method in terms of how to say, people have set up a set of criteria to evaluate how good a method is or what these methods should guarantee. So if we trace it back to the history, right, the most popular, one of the most well-known methods is called two-sample t-test from introductory courses. So this method was developed for a very simple reason, to compare a new way of beer brewing to a traditional way. So essentially, it's for quality assessment. You want to know whether this new way is actually different or better than the traditional way, or if the difference we see in the two ways of beer brewing is something meaningful, not just due to some measurement randomness. So because of this origin, I think about how these two things could be bridged in some meaningful way, because rigor was considered as important in statistics. But on the other hand, in genomics, we have so many options to do our data analysis. So one article I hope to introduce to our general audience is this very interesting review or perspective piece about 8 most important statistical ideas in the past 5 decades. And one of the ideas is related to what I will talk about today, bootstrapping and simulation-based inference. So permutation can be considered as a special case for this idea. So the idea is to do some resampling of your data to break some dependency that potentially exists between your variables. And after permutation, by disrupting such dependency, you can actually obtain a negative data set where the dependency is removed. So I think this can serve as a baseline for our data analysis, because then you can say whether the discoveries you observe in your data analysis is something meaningful. It's something that you cannot observe from permuted data. And how to permute the data? I'll give several examples in my talk today. And Pang will talk about one specific example, which I will briefly mention, and she will go into detail. So it depends on the set of variables you have, whether it's a supervised learning setting in which you have those features as predictors and a response you want to predict for, or it's unsupervised learning setting in which you don't have the response or outcome, but just the features. So for bulk RNA-seq data, I will consider it in this supervised learning case because here I can consider the samples belonging to more than one condition. And here the features for RNA-seq are genes. For the unsupervised learning, I'll use single-cell RNA-seq as an example. Here, the samples are cells and the features are genes. So the bulk example is more like a teaser to open up my talk to show you that if you use permutation, you may see something quite interesting. So this is a paper we published 3 years ago now, in which we pointed out that by popularity, you would choose certain method by following tutorials to do the differential expression or DE analysis. However, if your sample size is large enough, you may actually see that some of the assumptions you assume in the methods may not hold well in your data sets. And as a result, you may have more than what you expected in terms of false discoveries. And the interesting thing is that this was a coincidence finding, and it was a collaboration with Dr. Wei Li's lab, who is a computational biology lab, his postdoc who did the analysis, and my former student who was trained in statistics. So this is the major surprising result which motivated us to look further into the issue. So this is a data set from patients. So we have 51 patients pre-immunotherapy and 58 patients on immunotherapy, and all RNA-seq data of their blood samples. And DE-seq2 and EDGE-R are two of the most popular or widely used DE-seq methods. They have very good software implementations and very detailed tutorials you can follow. So the first finding that surprised us, but actually it's not a surprise because many people reported the same thing, but it's just that they didn't look into the issue as detailed. So basically, the two methods may not give you similar findings. So in this case, for this particular data set, the two popular methods give you different numbers of DE genes. EDGE-R twice as many as DE-seq2s. And the first question we want to ask is, which set of results should we trust more, right? And if you look at the overlap, it's not like DE-seq2 results are a subset of EDGE-Rs. So here the sample sizes are big enough for us to do permutation. Because if we think about the origin of these two methods, when they were developed more than 10 years ago, when RNA-seq, bulk RNA-seq was still considered a new technology, the sample sizes were quite small because they're expensive. So typically, if you run a wet lab, you may only be willing to just generate 3 bulk RNA-seq replicates per condition. Then it's 3 versus 3 comparison. And how can you get enough statistical power out of it? Then both methods have to assume a gene, every gene follows a parametric distribution under each condition. So specifically, both assumes that for count data like RNA-seq, every gene's counts follow a negative binomial distribution under each condition. So with this parametric assumption, they can have more power for finding the genes. So with just 3 data points as in their design, it's not possible to check how reliable negative binomial assumption holds because you just have to assume it. But with such large sample sizes, then we can check the distribution. But before I show that result, let's just first show the surprising result. That is by permutation, simple permutation, in which we randomly assign those 109 samples into 2 groups with the sizes kept as 51 and 58. But now because of random assignment, these 2 groups no longer make any sense. We just run each method again on the permuted data, and we can get a DE gene number. And we can repeat this for 1,000 times. We can have 1,000 DE gene numbers from permuted data. That will give us a distribution. And here, the height of the bar is the average number, and the standard error bar is just the standard error, standard deviation. And we can see that compared to the permuted data, from the original data, actually, the methods found even fewer D genes. This doesn't make any sense because we would expect fewer D genes from permuted data. So why is this happening, right? That's our first question. Why are so many genes found as DE from permuted data? And we check the negative binomial assumption, as I said earlier. The interesting thing is that for this data set, negative binomial does not seem to be a reasonable distribution. The reason is twofold. First of all, negative binomial can be theoretically justified if your bulk RNA-seq data are technical replicates, meaning that they are biologically identical, but because of sampling uncertainty, you don't have exactly the same number in each sample. So you have some differences. And that can be described by like Poisson distribution for sampling. And before Poisson, you can have a gamma distribution for the biological heterogeneity. However, here we are not having replicates. Here the 51 are patients and 58 are patients. And we know that there could be greater heterogeneity than replicates. That's one reason. And the second reason here is because we have enough data points to check how the distribution fits. And this is the commonly used quantile QQ plot, which can allow you to check whether a theoretical distribution fits data well. And because we see these points that do not lie on a straight line, we can conclude that here negative binomial distribution does not seem to hold well. And this is that was for HR, and this is for DESeq2. So we see the similar trend. So you may wonder, what will be the consequence if I didn't know this, but I just did the analysis? I used DESeq2 or HR to report my results. Here's what you will get from your gene ontology enrichment analysis. So you actually see these terms enriched in the genes you find with DESeq2 or HR. And many of those are actually related to immune response or immune process. So you may actually conclude that, yes, I can see the differences in immune responses between the two groups of patients because they are pre or after immunotherapy. So you may be very happy with their results. But if you actually know that, okay, these genes can also be frequently identified from permuted data, then you may step back and double think about the results. Probably it's just that immune genes are so dynamic that they can be easily found from the analysis anyway. So that could be one possibility, but that just opened door for more questions or critiques. So I'll just say that this teaser, even though we found this by coincidence and it was not a method paper, but somehow it received a lot of attention and discussion in the field because many people would think of bulk RNA-seq analysis as a done task. You have popular methods that have been used by many people, then there's no issue about considering using or how to change my analysis or use different methods. But we are just arguing that the data characteristics do matter. So essentially, which method to choose depend on your sample size and also depend on whether you can check the assumption of the methods, no matter how popular they are on your data. So for this data set, if we use this very standard Wilcoxon rank sum test, because this is okay for two sample comparison, if you don't want to adjust for other sample characteristics like covariates, you can use that. And at least on this data set, even though the results are disappointing because Wilcoxon does not give you any findings, but it's not self-conflicting. So you also don't see any discoveries from permuted data. So you don't have this conflicting results that bother us. All right, so that's the teaser part to show that permutation as a very simple task can reveal a lot of things, but we just need to just add it to our data analysis without too much trouble. Yes.
So just a quick question. So looking back at the initial plot, it seemed like there were two things that were there. That one was that the kind of confidence interval on the permutation was very large. But then the other was that in all of the examples shown, the kind of actual red box is far below the mean. So is that in a statistically significant manner that the red box is far below?
Okay. Yeah, actually, it's actually okay because this is just one standard deviation. So it's actually within one standard deviation. Yeah, I think the reason is that in this case, because you can see the two sample, right? So essentially, because of these outliers, you can see that if you move one outlier to another group, then the results will be very different. So that can explain why the numbers can change so large. Yeah. Thank you for the question.
Thanks.
Hi. Could you explain, like, so what does the distribution of gene expression look like in that human data? Like, is it just that there's a lot of like outliers,
It
or is
is.
there a lot of technical
I think
something
that's a
going
very
on?
important question. Yeah. So when you see those outliers, which are actually very large counts, so essentially looking at one gene, you will see larger counts than what you can explain using a negative binomial. And then the question is, and these are from patients, how do you look at those data? Do you think they are error measurements, or do you think they are potentially something meaningful? I think that's the question. Or maybe in this case, looking at just the mean may not be so meaningful, right? Probably you should go with like looking at the variance, or you should specifically focus on outliers. I think these are very important questions. But if we just run the DE analysis blindly, we'll miss those opportunities. Yeah, thank you. Thank you.
Did you check more data set? Because this is maybe this is only true for this data set.
Of course, if you look at our paper, we have multiple data sets. It's just that this one is the most striking one, and we just report. And we actually also tried this on TCGA data sets and GTAC data sets because both are from human populations. So they are patient data, not replicate data. So in short, I think the conclusion we want to draw people's attention to is that population, if you have data from human patients, you cannot treat them as replicates. And while the negative binomial assumption was justified only for replicates.
So this is more likely because the heterogeneity of the people.
Exactly, yeah.
Okay.
And these people may even have subpopulations, right? That you didn't divide them.
Yeah, another question is that, do you have any idea about if we can use a better distribution to model the count data rather than only using the negative binomial distribution in this case?
So there are, if you look at the literature, there are more than 100 DE methods. Many of them didn't become popular because every method, but as long as they were published, right? So they have their reason, they have their underlying model assumptions. Some of them, as you mentioned, they will go with some non-parametric modeling for each gene's distribution. But the question is, if the distribution is something not like negative binomial or not something you can easily describe by a parametric model, whether the mean parameter is still your primary interest. I think that's the question. Yeah, so I think we need to think about what we are really looking for. Because for negative binomial, people think, okay, the mean parameter means the biological expression or the true expression we are hoping to infer. That's why they go with the differential expression, right? But as I said earlier, I think it really depends on the data. And also, I think during the discussion, online discussion, some people did show that if you remove those outliers, you can make negative binomial fit well. And then you can proceed with your normal analysis. But here, I think we need to think about the data as patients, right? So every data point should be valuable in some way, unless we made a mistake in the measurement. So how do we consider the outliers? Yeah, but that's a very good point. Yeah, thank you. So that's why when we use the Wilcoxon rank sum test, essentially we drop the parametric assumption. We're just comparing the two sets of numbers in terms of ranks. All right, so now I will move on to the unsupervised learning case with single-cell RNA-seq data. Samples are cells, features are genes. And I will talk about two examples. And first one about visualization. Second one about this metacell practice. And Pan will, because Pan is the first author for the metacell mcRigor, standing for metacell rigor, and she will give detailed discussion on this. So I'll be brief on this, but I'll talk about how we use permutation to help us tune the hyperparameters of t-SNE or UMAP for better visualization. So single cell visualization is a commonly used way to show the data, right? Because we know biological papers have figures and they want to look at the data. People want to look at the data. But for single cell data, the challenge is that every cell is represented as a high dimensional gene expression vector. Like depending on how many genes you measure, if it's 1,000, then every cell is 1,000 dimensional. How can you see that high dimensional space? We don't know. So two-dimensional space is what our human eyes are most comfortable with, or the papers are printed as 2D. So that's why the very, hopeful thing is that we can see the cells in a 2D plot and use the plot to have some idea about their relationships. For example, people want to see clusters representing cell types. They want to see trajectories representing cells' continuous changing process. But t-SNE, which was actually introduced in 2008, at that time, there was no single cell data, but it was introduced as a general visualization technology of high-dimensional data. It was designed to encourage similar data points to be clustered together so you can see clearer clusters. But there is a key hyperparameter called perplexity. Perplexity essentially defines the size of a neighborhood. If it's very small, then you will have smaller but many smaller clusters. If it's big, then you will have a big cluster. So looking at this whole example in which the original data is just two-dimensional, so this is considered the true visualization. If you run t-SNE from a small perplexity to a large perplexity, you can see very different visualizations. Sometimes you will see the two clusters, sometimes you don't. And also, if you want to use the visualization to say anything about whether two clusters are close to each other or very different, then the distance may not mean anything. And this critique has been also reported by this commentary article on Nature Methods about using t-SNE or UMAP to see the data. And the last sentence here is that the abstract is sometimes these methods take a second thought. So our method, the one we propose, kind of like a compromise. So we know that there are voices against using them for visualizing our data. However, we think it's very hard to discourage or let people say, okay, don't use it anymore because they have been in every paper, right? So if we want to use them anyway, is there a way to use them better? So to answer this question, we actually formulated the problem as a question as, okay, is a cell's embedding dubious or trustworthy? How can we define this? How can we know if we can trust a cell's relative location in the figure, in the 2D figure? And our idea is to compare the cell's neighbors before we do the embedding, which means that in the input data before t-SNE or UMAP, and after the embedding, which means the 2D figure we see. And we title our method, we call our method scDEED for answering this question. And our intuition is to look at what we call mid-range neighbors of a cell. So this one, the purple one, cell 1, is of our interest, our target. And we look at the mid-range neighbors, and we define that as half of all cells in our definition. But this is something we can change. However, we think this is a reasonable proportion. So if the neighborhood is so small, we know both t-SNE and UMAP can do very well in preserving the neighbors because that's in their algorithm for encouraging the close neighbors to be preserved. But we are looking at the mid-range neighbors since we are interested in between cluster relationships. So not just the local neighborhoods. So in this case, we need to look at a bigger range. And also, how do we define pre-embedding neighbors? So in practice, when people do single cell data visualization, the pre-embedding neighbors or the pre-embedding space is not the original high-dimensional space, but what people did after PCA. So essentially, PCA is commonly performed as a pre-processing step to reduce the dimensions from 1,000 or thousands to 20 to 50 PCs. The hope is that PCA removes noise but preserves the major important signals. But still, 20 to 50 PCs are not enough for visualization. That's why we are not good for visualization. That's why we are using the t-SNE UMAP to project them from the PC space to the 2D space. So in this case, in the PC space, cell-cell distances are measured by Euclidean distance, and that's how we define the pre-embedding neighbors. And this is exactly what's used in the t-SNE or UMAP algorithm, the Euclidean distance in the input space. While in the 2D space, we are also using the Euclidean distance we see in this figure because that's how our human mind or eyes interpret this figure. So we want to compare these two sets of neighbors, each chosen from each space. And we want to compare their ordered distances. So essentially, what we are doing here is that we are taking each set, preserve their order in their original space. So this one means that the neighbors are ordered by Euclidean distances from the closest to the farthest in this half range and in this PC space. And we have another ordered set of neighbors in the 2D space. And for each set, given the order, we actually record the 2D Euclidean distance of each neighboring cell to the target cell. So in this case, the two vectors we construct are taken from the same 2D space because that's how we interpret the figure, right? So that's what we're looking at. And then we compare the consistency of these two sets of ordered distances. And we use the Pearson correlation as the measure for consistency and define that as a reliability score. The reason we choose Pearson correlation is because it actually considers the actual distance values, not just the ranking. Because for example, here, this one is very far away, but these are not so far away. So when we look at the figure, we're not just interpreting the relative ranking, but actually the actual distance. So that's how we define the reliability score. And this cell 1, is considered by us to have a good reliability score, while cell 2 is something we consider to be bad or dubious. So in this case, if you see the figure, the pre-embedding neighbors, looking at them on the 2D space, you can see that they are very scattered, right? So they're not concentrated, or they don't look like the neighbors to cell 2. So this means that t-SNE or UMAP embedding changed the half-range neighbors a lot after we do the projection. So in this case, the reliability score is considered low. So that's the intuition. And our intuition turned into this score definition. But we still need some calibration, or we need some baseline. And here the null hypothesis using the statistical language or the baseline in the scientific language is that here, what will happen if the cell's neighbors are not meaningful? How can we create such a scenario in which the mid-range neighbors are just random, right? How can we do that? And in this case, we think about permutation. So if we think about permuting each gene randomly across all cells, we do that independently for all genes, what will happen? So you will see that by such a permutation, you did preserve every gene's marginal distribution, one-dimensional distribution, because they are the same values, just differently ordered. But what you disrupt is gene-gene correlations and also cell-cell relationships. Because in this case, your permuted cells do not correspond to the original cells. They are a new sample, and this new sample comes from this new distribution. But since these permuted cells, if you think about them mathematically, they're actually exchangeable with each other. So therefore, each permuted cells can be considered neighbors to any other permuted cells, right? So you don't have any meaningful biological relationships. And with such data, if we apply our score calculation to each permuted cell, we can get a score. But this score comes from the null case in which that you don't have meaningful neighborhood preservation. And we can get one null score from each permuted cell, and that will give us a distribution. And this distribution can let us calibrate the scores we calculate from our real data, from our original cells, whether they're high enough. They must be high enough to be called trustworthy, right? They should be much higher than most of the null scores. But regarding the dubious definition, how do we set a threshold? So here in our paper, we set a threshold to be very, very conservative in the sense that if you are worse than the majority of the null scores, then you are really, really bad. And then we flag that score as dubious. But you may actually change this. You may actually say, okay, I don't want to be too conservative, and this threshold is adjustable. But regardless, here, this distribution just gives you a way to calibrate the scores for your real cells to see whether they are good enough. And also, I want to clarify that here, the permutation only needs to be done once. Because once we do this permutation once for the whole matrix, we can get a score for each permuted cell, and that pulling them together, we can get a distribution. So we don't have to worry about the computational cost for many rounds of permutation. So let me just show you what we find. So this is one figure from a 2019 science paper about a species called Hydra in the ocean for their single-cell RNA-seq data measured for the first time in history. So you can see that this is the original t-SNE plot using the default perplexity 40 in SURAT. And if we run our dubious detector to detecting dubious embeddings, you can see that to our interest, very interestingly, a lot of the dubious embeddings are in those small clusters here. And so we just wonder whether because of this, distortion by the 2D embedding, we see these small clusters, but they shouldn't be as so isolated or standalone as we see in the figure. And in fact, that's the case, because if you go back to the labels, you see that a lot of the labels of the clusters on t-SNE were given a number, and the number meaning that they share with the same cell type label with some other clusters, but because they look like separate clusters, they were given different numbers, but that may not be biological. So one example you can see here is in the original figure, you will have the 3 small clusters surrounding the big orange cluster, and the orange cluster represents stem cells, while the 3 small clusters have the same label, neuron ectoderm cells. And with our optimized visualization, so how we do that, we can actually change perplexity, calculate the number of dubious embeddings, and find the perplexity value at which the dubious embeddings are minimized. So at this perplexity, which is larger, we can see that the 3 small clusters are now unified down here, and they become far away from the big cluster representing stem cells. And this representation has better agreement with what we see from the gene expression data in the heat map. So I think this is just one example to show that the way visualization is plotted matters, right? It will change our perception of the relationship of cell types if we look at the figure closely. Another application, I'll just show what result in our paper. There are many, but I'm just showing a very small set of that, is this consistency between t-SNE and UMAP. So since UMAP was popular, the belief is that it better preserves the global topology or relationship of cells compared to t-SNE. And indeed, as we found in our result, UMAP has 2 hyperparameters, the minimum distance and the number of neighbors. So with the 2 hyperparameters together, we can see that the UMAP results are more stable than t-SNE results. So t-SNE results can change more dramatically as you change perplexity. The UMAP turns to be more stable. However, the other thing we note is that if we use our approach to minimize number of dubious embeddings by changing the hyperparameters of either method, then we can see that with the optimized visualization, their consistency becomes better than if we go with the original or default. So that's another indirect evidence to show that the optimization is meaningful. So with the time constraint, I would just say that conclude this part for visualization. And for the metacell part, I'll just briefly mention the problem, and I'll give the time to Pam for her talk. So what is a metacell? It is a common practice used in the single cell analysis field.
Go on.
Sure.
Quick question about your previous thing. So are you actually making the case to use PCA instead of t-SNE or UMAP?
No, not at all. So PCA here is just using as a pre-processing step for reducing dimensions from say 1,000 to 20 to 50. But it's not used as a visualization tool because if you just look at PC1 or PC2, in most cases, you cannot see clusters.
Okay.
Yeah.
So you still recommend using t-SNE or UMAP?
I wouldn't make a point for that. I would say it's what we propose for the method we call scDEED is only when you want to use UMAP or t-SNE anyway. And we think that using that's better than not using it. That's our point. But whether you should completely remove t-SNE or UMAP from single cell papers, I think that's a separate point.
Okay. Thank you.
Thank you. Yeah.
I thank you for the wonderful method. So I wonder, in the case that if I want to visualize the cell from different tissue, for example, and a different batch, would you suggest to do the permutation within each cell or each batch, or would you consider doing them jointly as a whole permutation?
That's a very good question. But I think also, I always want to ask your goal. What is your goal of doing this visualization? What do you want to see in your data, right?
Yes.
Yeah.
I honestly, so yeah, maybe I just in a data-driven approach. So maybe, I previously do not have a very clear goal about what I want.
You just want to look at the data, right?
Yes.
So in this case, I would say if I were you, I would do both ways. Because I will say, if you look at all the batches together, then you can see where they are, right, on the UMAP or t-SNE. And also, if you want to look at each batch separately, then you can see whether the together plot or the joint plot was distorted in some way. I think I'll just try multiple ways to visualize the data, including PCA, right? So, and try to do exploration. Yeah,
thank you.
That's what I would suggest. But on the other hand, it's very important to know whether you want to preserve the cell types or you want to, how do you interpret the batch effects? Are they technical effects you want to remove, or do they also contain possibly biological effects you want to preserve, right? That's also a question, yeah.
Hi, this is really interesting. I wanted to clarify, it sounds like you're trying to perceive the visualization preserves Euclidean distances based on gene expression. I wonder if you thought about maybe considering manifold distances or geodesic distances, since a lot of people in the community think that that's what we're trying to preserve in the visualization.
Yeah, thank you. So actually, this is another thing. So the Euclidean distances, we are considering them in two spaces. I want to clarify further. The first one, going back here, is the Euclidean distance in the PC space. So that is not, so basically there's a belief underlying, that is, once you project your data from say log transform or log normalized gene expression in the original space, gene expression space, to the PC space preserves the biological signal, removes the technical noise. That's your belief. So in this case, Euclidean distance in the PC space is considered to be okay. But when people say manifold distance, they are thinking that there is an even lower dimension than the PC dimension. And they want to use the manifold to describe that. And that is why when they use that idea or assumption to get the visualization, you will get a different visualization from t-SNE or UMAP. But since here we're just looking at t-SNE or UMAP, and in their 2D space, people are still interpreting, or at least our eyes are reading 2D Euclidean distance. Because we're thinking about clustering structure, right? The round clusters, and that is a Euclidean distance-based idea. So that's why in this case, we're considering Euclidean distance in 2D versus Euclidean distance in the PC space. And manifold is not part of the discussion. Yeah, that is the clarification.
Yeah, just a follow-up question.
Yeah.
So I just heard you mention like doing PCH is for removing the noise beforehand, but has your group just looked at the original data and kind of either Euclidean distance or cosine, or like how does it compare with this? Like,
so could you say your question again?
Also, has your group looked into the just distance between within the original data before even PCA?
Oh, actually, that is another mathematical fact, is that if you have such a high dimensional space, like 1000 dimensional space, Euclidean distance is no longer meaningful. Yeah, that's the reason why we didn't look at Euclidean distance within the original space.
I see.
Yeah.
Okay, thanks.
Thank you. Yeah. All right, thank you so much. Wow, that's a very good discussion. And then I'll just be quick for the second part. I'll just say, because Pam will introduce that in detail, I'll just say that metacell is a common practice in the field because of sparsity. So people say that, okay, especially if you look at single cell multiome data, for example, RNA-seq versus ATAC-seq, it is known that the data is very sparse, right? Many of the genes or enhancers were 0 in the data. So how do you make sure that you can still do something meaningful to find something in your analysis? Metacell is a heuristic solution in which people just try to group cells into very small clusters, right? And hopefully each cluster represents the same biological entity as the original cells. Then you look at those small clusters, you take the average of each feature in the cluster, and you call those metacell profiles, and then you analyze those data instead of the original data. And here the goal is that you can be faster because there are fewer metacells than original cells, and also metacells are less sparse. But the question we want to address here is, what is a reasonable definition for metacells? Because you can have them very, very small, or you can have them bigger. But of course, there must be some underlying, how to say, sweet spot or trade-off in the middle you want to find. Because you can think about two extremes, aggregating all cells into one metacell, and that is what people call pseudo-bulk. So you're not having single cell resolution. Or the other extreme is that you don't do metacell at all. You just keep single cells. So where should we stop? So I'll just say that the motivation for, we want to come up with a statistical definition again. So our definition actually is based on this two-layer model from the literature Pan will talk about, in which we believe that the biological variability can be described by what we call an expression model. And given the biological expression, there is a measurement model that performs the sampling, which is usually captured by Poisson sampling to give us the count. So if we believe that the cells should be merged into a metacell, then their differences should be only due to the measurement model, and they should be largely identical in terms of biological expression. And because of this two-layer assumption, we can define a metacell and giving it a statistical concept. And one notable thing about the measurement model is because it's only a sampling process, and you already have the biological counts of RNAs. You're just sampling during the sequencing experiment. Then we can reasonably assume that gene-gene correlations are not induced by measurement model, but only comes from expression model. So this can give us an idea to construct a statistic in which we can measure how dubious each metacell is based on gene-gene correlation. So the idea is, if the metacell only contains single cells that differ based on the measurement model, then gene-gene correlations within the metacell should be minimal. So that is how we design a statistic, and we call that MCDIV. So Pan will give more specifics about this. I will just go back to my theme to say how permutation can be helpful for calibrating the values of the statistics. So we can use that null distribution to define a threshold, so we can call a metacell as dubious or trustworthy. So coming back to permutation, in scDEED, we did within gene permutation, and we preserved our marginal distributions of each gene, but we made genes uncorrelated. And furthermore, if you look at the permuted cells, they do not correspond to the original cells. The cell library sizes in the original cells are gone. But if we go back to the two-layer model, which Pan again she will detail, here is that the cell library sizes is very important, because it depends on how many counts you receive each cell, and that total count is divided into genes. And because the total is the same, then the genes within the cell will have some small correlations just due to this multinomial sampling, right? To have some dependence among the genes. But we need to model that, and we need to capture that in the cell library size. So how can we do that? So we know that just doing the within gene permutation, yes, we can make genes uncorrelated, as we believe that should happen within a trustworthy metacell. But removing cell library sizes will make this not a valid null for our statistic. So to account for that, Pan came up with this smart idea of permuting within each cell. So in this case, we can actually preserve the cell every row sum, or the total of the cell library size for each cell. So you can see that here we preserve the cell library sizes, but we change the genes. Since you're randomly permuting the genes within the cell, we call those permuted genes, and they do not correspond to the original genes. So even though this can give us a null case in which the cell library sizes are preserved, but because the genes are different, we still miss something. So then we did a normalization. So the normalization is by removing the differences of genes. So we would have a normalization factor for the original data by doing within gene permutation. So this creates an uncorrelated case in which the genes are the same original genes. And we do the same thing for the permuted matrix. So here after we do the permutation within a cell, we further do the permutation within each permuted gene, and we call this double permutation. So this can give us a normalization factor to account for the fact that here the permuted genes are not the original genes. So each set of genes have their corresponding normalization factor, and together we get a test statistics null case. So this is an idea which we think is very interesting, and it turns out to work pretty well. So without specifying the detail of the statistic definition, I'll just say that what we did procedure-wise is to perform double permutations for each metacell. So we just need to do this double permutation again for once. We get a null value for each metacell. So you can see that as a result, we'll have a statistic for each metacell and the corresponding null statistic value for each metacell. And pulling together, we want to use the null values to calibrate a threshold for our actual statistical values. And what we are doing here is that we observe the null values have some relationship with the metacell size. And here the size means the number of single cells contained in each metacell. And this can give us a size-specific threshold based on the null values. And given the null values, we can consider each original metacell's statistic. Whether it's too high above the threshold, we call that dubious, or if they're too low, or they're low enough below the threshold, we call them trustworthy. So again, we try to use permutation to derive a threshold. Well, and again, like scDEED, we only did permutation once. So I would just say that for the permutation part, I will talk about here. So basically, bulk DE as a teaser, right? Here it's a simple thing. You just permute the condition labels. And for single cell, we consider within gene permutation or double permutation, because for the former, we just want to make sure that the permuted cells are like exchangeable, a random sample without any biological relationships among the cells. And for the double permutation, we did it because we want to preserve the cell library sizes first. So for this part, I want to acknowledge my collaborators, both from the computational biology side and from statistics side. Lucy, she's a statistician, and also, of course, Han for mcRigor. But before I end my talk, I just want to briefly say that this idea of generating some null data is beyond permutation. Permutation can be considered as a special case for this concept we hope to promote. We call that synthetic null data. It means that we generate data from the null hypothesis, and we can use that as a calibration device for our original data. So one example I want to point to is that we propose this method called cluster DE, which is still a preprint, but we are making it more and more comprehensive as time goes. It's a post-clustering differential expression method. We showed it's working for both single cell and spatial transcriptomics data. And furthermore, we're planning to add more to it to other omics to show it's more general. So the idea, briefly, is that we propose to generate synthetic null data that represents a homogeneous cluster. But in other aspects, we try to mimic the real data, the original data. So the hope is that if you perform clustering to identify clusters as potential subpopulations, and then you compare the two clusters to find DE genes as markers, we hope to avoid this over-clustering bias. In other words, even though your original data is just one homogeneous cluster, you may still divide it and then even to have two clusters, and your DE results can still show many, many marker genes. This is something we observed in SURAT applied to single cell line data. You can see that. So we try to remove those biases and also to prioritize the true markers if the two cell clusters are indeed distinct. We use this negative control. And I'll just be brief by showing that it's working by prioritizing like true marker genes in like the CD16 in a comparison of two monocyte subtypes, CD16 versus CD14, and pushing down housekeeping genes down the rank list in DE, because housekeeping genes are typically highly variable with large variances, and they can actually drive the clustering or be involved in the clustering, and as a result, be identified by the DE step between two clusters. But by setting up a negative control, as in our synthetic null data concept, we can push down the ranking of the housekeeping genes, but preserving the high ranking, top ranking of the true marker genes. And this gene set enrichment analysis showing that, yes, the known markers are ranked highly in our DE list, but housekeeping genes are ranked much lowly compared to what SURAT gave us. And the reason for that is because we have this contrast. So we can have a target D set of D scores and the null set of D scores. We do the comparison, and only the genes with a large contrast will be high in contrast score. And we show that the marker genes are higher than the housekeeping genes. And also apply to the clustering result, this approach can help us merge spurious clusters that are not so well separated. We can decide to merge them. And this can also show us that if we use the idea, then if you run direct clustering with different resolutions, like the high resolution will give you smaller, more clusters, while low resolution give you fewer, coarser clusters. We can apply this idea to each branch to decide whether they should be merged before we call DE genes. And we can see that we can make the two sets of clustering results more consistent by finding the same cell types from the PBMC data where people know the best. And apply to spatial data, I'll be quick by saying it's the similar post-clustering DE issue. You use spatial clustering to find regions and you want to find markers. So our idea is that if you really believe a spatial cluster is something meaningful or distinct, you should have a very clear boundary between the adjacent cluster. But we can design negative control or synthetic null data by making the boundary smoothened. Then you don't have the sharp changes in G expression, but otherwise you have the same overall trend from high to low or low to high, but just no sharp boundary. And with this negative control, then you can see that this is what we can find. So for your spatial clusters, the marker genes we find, we nominate will be much sharper markers compared to what you find from SURAT because we set up the negative control. So in short, I'll just say that this is a general approach to set up synthetic control for removing cluster-induced bias. And beyond these two single-cell spatial omics, we actually showed that it works for other things, right? For other types of omics, like including population-based bulk RNA-seq and also microbiome data. So these new results will be added. And finally, I'll say that cluster DE can further be generalized into this approach called null strap. It's led by my postdoc Chang Hu, who actually generalized this to the variable selection problem in the high-dimensional prediction model. So this is a very, very general question, which, okay, if you have many, many predictors or features, which are the ones that are most predictable of your response? So, and our hope is to also achieve the false discovery control. So not just variable selection, but some guarantee. Before our work, in the statistical literature, there are methods called Model X knockoff or multiple data splitting for FDR control. And there was a Nature Biotech paper, if you are interested, it's called Stable. It was published actually last year, and it uses the Model X knockoff with multiple runs and aggregation. So it's like a stable version of knockoff. And we can see that the issue with existing approaches is that they often have low power. So all these approaches depend on random seeds. And we can see that across many random seeds, you will have zero discoveries, no features found, and that could be an issue. But using null strap, we can have a much higher power because we preserve the original data. We didn't change the data. We just have a parallel synthetic negative control, and we can have feature selective with good prediction power. And to our surprise and pleasure, there was an independent benchmark just posted a few weeks ago, which compared our null strap, which is still unpublished, to this published stable method. And in this independent benchmark, we can see that they found in terms of performance like AUPRC, precision recall curve, and the classification F1 score for true features versus null features, we are the best. Even compared with the common approaches like mutual information and random forest for variable selection, we are better. And notably, we are very fast. So we are even faster than the mutual information in terms of computational time, so which gives us a lot of confidence. So I'll stop here by saying that my overall goal, the overall goal in my research group is toward principled inference. And in particular, I think this is very, very challenging for unsupervised learning because you don't have a very clear prediction goal. And there are many, many issues I can touch about, including what is considered reasonable inference, conditional or unconditional. But this synthetic control setup from the beginning is for unconditional. So we want to consider what is the bias introduced by the whole procedure, including clustering and also DE test. And Bootstrap is something we really draw the inspiration from Bootstrap. So we're thinking about using resampled or synthetic data, but we're sampling from a null hypothesis or null model instead of just the original data. So our outlook is that we think there could be more potential applications. So that's why I squeezed some time at the end of my talk to introduce it. So, and this is the slide for the second part. Yeah, thank you very much for the primer. For listening to my primer, I should say, yeah. But I understand for the second part was very fast, so I didn't have time to go into details. But the permutation is the primary part. Yeah.
I think that there were a lot of questions during, but if anyone has any questions after the talk.
All right.
I think let's break for like 5 minutes or so, and then we'll be back for the
Thank you. Yeah.
talk. Thanks.
Thank you.
Great. Okay. So thanks everyone for sticking around. So now we're going to have Anne give her talk on mcRigor, which I mistakenly read as mcRigor the first time I was reading this. So statistical method to enhance the rigor of metacell partitioning and single cell data analysis. So thanks everyone.
So thank you for the introduction. So my name is Pan, and I'm now a postdoctoral researcher working with Professor Jessica Li at the Fred Hutchinson Cancer Center. But previously I worked at UCLA, and this work was mostly performed when I was at UCLA. In today's talk, I want to further elaborate on this work called mcRigor, which is exactly an example of how to use permutation to serve as a negative control to make our analysis more rigorous. And as introduced by Jessica previously, this work aims at enhancing the rigor of metacell partitioning in single cell data analysis. And here we do call this method mcRigor instead of mcRigor. Here MC stands for metacell. And let's just start with a quick recap of what is a metacell in case that some people still don't get it. So here, this figure is from a very nicely written review paper published in Molecular System Biology last year. Right, so this figure is from a very nicely written review paper about the metacell method. And so on this top left panel here, this is a typical format of a single cell RNA sequencing data, which is normally just a matrix with cells for columns and genes for rows. And we know that in this kind of data matrix, there could be many zeros, maybe like as high as 90%. And many of them are due to the imperfection of single cell sequencing technology. Because this kind of technology is very high throughput and we can have very low per single cell sensitivity, which means that for many genes, these genes may be expressed in the cell, but their expression can be just not captured. So intuitively, we do not want like all these zeros in our downstream analysis. That would cause a huge problem. Or to say we want to solve this kind of sparsity issue. And the metacell method is exactly a pre-processing step in single cell data analysis developed mainly to solve this kind of sparsity issue. So what metacell method does is that it first tries to identify similar single cells shown as the colored dot here within one black circle. And then we just aggregate all these similar single cells like by averaging. So here, each black circle, or you can also think of it as a small cluster represent one metacell. Then we just need to define like an average profile for each circle. And then we can end up with this gene by metacell matrix, and we will proceed. We'll proceed from here with all other downstream analysis tasks. And by doing so, we can reduce the sparsity level to approximately like 50%. But at the same time, we are also paying some prices. The main price is that now we have a much smaller number of analysis units or observations. We have metacells instead of single cells. So that means we have lower resolution. But there are also some cases where people think that having a smaller number of analysis units is not always the bad thing, because that also means we have like better scalability, right? So, so due to all these reasons, This metacell idea has been pretty popular in single-cell data analysis, and it has been adopted in many high-impact studies, as I will show in short. But the question that motivated our research is, what is a reasonable definition for a metacell? Like, because if you think of two extremes, like mentioned by Jessica, the two extreme is like, for one extreme, we can just aggregate all the single cells into one metacell, then we just totally lose the information. Or on the other extreme, if we just leave the single cells as they are, then we are not doing any aggregations. So we think there must be some sweet spot in the middle that could balance these two extremes, and we cannot just do it arbitrarily. So we wish to answer this question, like how can we find this sweet spot? And then this slide lists the existing use of metacell idea. There are 5 general methods, starting from, and these general methods, by general method, I mean that these methods have been developed into software packages where people can just directly use them. So start from the very first version, the metacell method, which proposed the metacell idea, and the second version, metacell 2, by the same research group, and we also have supercells, C cells, and also the most recent metaQ, which was published just this year. And besides these 5 general methods, we also found that this metacell idea has been used by many in-house data analysis published in very high-impact journals like Nature. They have either come up with a more straightforward way, like just maybe k-nearest neighbors, to define metacells, or they just use one of the 5 general methods. But looking at all these papers, we do not really see a consensus on metacell definition. People can just do it in their own way. So, motivated by this problem, we develop our own method, mcRigor, because we wish to improve the rigor of metacell definition. And generally, we wish to answer 3 questions. The first question is how to define a metacell. And the second is how to find the dubious metacells generated by existing metacell methods that do not follow the appropriate definition. And the third question is, how do we optimize metacell partitioning for a given data set? And just in short, our method is designed as a way to help with existing methods to make them more rigorous and obtain better metacell partitioning results, but it is not like a completely new method to build metacells from scratch. So it is more like an add-on concept to make existing methods more rigorous. Okay, so the first question, like how to define a metacell. So to answer this question, we go back to the very first publication, the one for metacell method, that proposed this metacell concept. So in that paper, a metacell was defined as a homogeneous collection of single cell profiles that could have been resampled from the same original cell. Well, this is a biological interpretation or definition, and we want to translate it into something statistical to work with it. So our translation is that given this concept, if we see variations within a metacell, then we believe these variations within the metacell should be attributed exclusively to measurements or technical error instead of true biological differences. So given this translation, how do we actually address the problem? Well, I think now the question becomes, how do we define this measurement error in single cell sequencing data? Because we believe a metacell should, single cells within metacell should be varied only by these measurement errors. Well, we actually use this paper's formulation to answer this question. This paper is a very nicely written paper about single cell statistical modeling. The authors of this paper separated the single cell model into 2 layers, an expression model layer and a measurement model layer. And these 2 layers, they together give us the model that we can obtain our observed count eventually. So more specifically, an expression model describes the feature's true gene expression in the cell. And the measurement model is literally like a conditional distribution. That is like, given the genes true expression, what are the observed count we can get in our data? And we formulate this two-layer model into statistical language. Here we consider the N cells as N observations and the P genes as P features. And just to briefly mention that our method can also deal with other data type like SC ATAC-seq data. So in that kind of data, the features are the peaks or the chromatin accessibility. So that is why we generally call it features instead of just genes. Okay, so the first layer is the expression model. The expression model is about the true gene expression in cell I, represented by lambda I here. And this lambda I is a p-dimensional statistics, is a p-dimensional vector where each dimension represents one feature or one gene, because we usually look at these features, these p features jointly. And the expression of these p features are assumed to follow this F distribution about the biological state of the cell I, which is represented by xi. So examples of this biological state information could come from things like cell types or pseudotypes in a lineage trajectory. And I also want to point out that this lambda here, it represents the relative expression level with its components summing up to 1. The reason why we focus on this relative expression instead of the absolute expression is because we believe the single cell sequencing technology can only provide like reliable relative expression information, but not absolute abundance. We believe those absolute abundance information are kind of lost in the sequencing procedure. And then for the second layer is that given this lambda I, the measurement model depicts the variations or the errors introduced in the sequencing procedure. Like given the true expression lambda, how do we end up getting the observed count Yij? And so this kind of procedure is usually assumed to be somehow similar to the sampling procedure. So that is why we usually just use the Poisson distribution to model this layer. And on this right-hand side, the Ci is the expectation of the cell library size, which is Yi plus. And it is also called the per-cell sequencing depth sometimes. So based on this two-layer model, our statistical definition of the metacell is a group of single cells that share the same lambda. So the cells in the same metacell must have the same lambda, but they can have like different Ys. And then we can formulate this into a statistical question. Like, can we check if an existing metacell satisfies this definition? If yes, we can just call this metacell a trustworthy metacell, and otherwise we call it a dubious metacell. So before we dive into our own method, just some more motivation of why we want to identify dubious metacells and why is it so important. So this is again a figure from that metacell method review paper. Here, these single cells are colored by their cell state. So each in this bottom left panel, each black circle represents a metacell. And so pretty obvious is that this middle metacell is dubious because it is a mixture of cells from two different cell states. And if we are interested in studying the association between two genes, like gene A and gene B here, for example, which is a very common and important analysis task for single cell data analysis. If we want to study this kind of association, we can first look at their association at the single cell level. And at the single cell level, you can see that actually these two genes, they do not have much association, at least like no positive association for sure. But if we compute their association or correlation at the metacell level with one dubious metacell, then this dubious one will lie here in the middle, and we may get a false positive association because of this single dubious metacell. So this is an example of how bias can be introduced by the dubious metacell, and that is exactly why we care about those so much, and we want to avoid that. So to avoid these biases being carried down to a downstream analysis, we propose our method mcRigor, which generally gives a statistical criterion to first identify the dubious metacells consisting of cells from different cell states. And also after identifying the dubious metacells, the next step could be to nominate the top performing metacell methods and also optimize its hyperparameter. And here we mainly focus on the granularity level hyperparameter gamma, which is essentially like the average number of single cells within one metacell. Or you can think of it as the metacell sizes on average. And we hope to choose this gamma in a data-specific way. And so at the most basic level, we use a feature-feature correlation metric as a way to help us build this criterion. And the intuition has already been mentioned by Jessica, I think, which is that since we can consider the measurement process as somehow similar to the sampling process, we can think of it as like the sampling for each genes are somehow independent. So for a trustworthy metacell, its feature-feature correlation matrix should be something similar to the bottom left matrix, something similar to an identity matrix. And on contrast, for a dubious metacell, its feature-feature correlation matrix can usually demonstrate this kind of clear block structure. So in order to make use of this intuition, what we do specifically is that first we need to build a per metacell statistics that can describe how much likely is this metacell dubious. And well, this per metacell statistics is called metacell divergence, or MCDIV. It is built based on the feature-feature correlation, as I just mentioned. And also we will need to build construct a null for these statistics, because we will need this null to evaluate the significance of the statistics. And as Jessica previously mentioned, we use a permutation-based strategy to construct the null. And an important point that we need to consider is that in this null construction, we want to preserve the cell library sizes. And the reason for this is that this Poisson distribution, or this measurement model, is equivalent to the joint multinomial distribution. And this cell library sizes Ci and Yi plus is involved in this multinomial distribution. And we know that this sum total, this cell library sizes, can introduce a small amount of feature-feature correlation into this data, and we need to account for that. So that is why we need to design this very unique double permutation strategy to make sure that we disturb.
Can I ask a question?
Yeah, sure.
On the previous slide, so like, how do you calculate the correlations first of all? Because there's like mostly zeros, and then
yeah,
like which type of correlation metric do you use? And then I think, can you just go to the previous slide?
Okay.
Sorry. And then what prevents like this sort of I guess criteria about the correlation? Like it seems like the simplest way then to solve, like to get, you know, not these sort of off diagonal things would be to have like very small metacells, like 2 or 3, because then you don't have these off diagonal sort of doublet metacells showing up. Like what is the counterbalance to like actually make metacells that have lots of cells in them? Because it feels like just the simplest way to get a trustworthy one would be to have like very few cells in the metacell, because then you're not going to have these correlations between cells that don't belong together.
Yes, exactly. That is a very good question. And to answer your first part of the question, we actually simply use the Pearson correlation to calculate this feature-feature correlation. And the reason for that is because we have this null construction. And in the null, we also use this Pearson correlation. So although there are many zeros there, in the null, since we use the same kind of correlation, you can still control for that. And actually, I have also tried the Spearman correlation, which may handle the ties better. And it turns out that that correlation gives similar results. So we eventually still use this Pearson correlation. And it is a very good point that you mentioned for those metacells with very few single cells, like 2 to 3. For those, I mean, like if the metacell is that small, then they are usually trustworthy. So that is why I think in our correlation, it doesn't really matter if the metacell with 2 or 3 cells you can get a very reliable correlation estimate, because those metacells are already small and we believe they are very trustworthy. And I think usually for a dubious metacell, the size of them usually starts from at least like 7, 8, or 10, something like this. And for those, we can get a pretty good correlation estimate.
The metacells are all the same size, or is it like kind of somewhat cluster specific? It's not tuned for the cluster that they end up falling in. They're all the same size.
Right. So this granularity level parameter is actually like the average size of all the metacells. And this is only the average size, and actually the size of those metacells could vary. They can vary like maybe from 2 to 3 to even 50 or 100. And how the size distributes across all the metacells, that thing kind of depends on the metacell method, the general metacell method that has been previously designed. So, and we are actually also working on like developing a new metacell method that could incorporate our considerations, but that work is still ongoing.
Hello, I have a question. Why do you think a good metacell should have like non-correlated features within this metacell? I don't quite understand this conclusion.
Okay, so right, that is like the key claim in our paper. And the reason for that is because we believe the single cells within a trustworthy metacell or within a good metacell should be at the same biological state. So the variations in the expression of those genes should be exclusively due to the measurement error. And by measurement error, we usually mean the sampling error. And in the sequencing procedure, we usually just sample those transcripts from like the whole library pool of these transcripts. And we think that procedure happens independently for each gene, that sampling procedure. So we think for measurement error, the gene correlation should not exist.
Okay, the second question is about single cell ATAC-seq. As we know, the single cell ATAC-seq is even sparser than the RNA-seq, and many elements are even 0 or 1. So in these cases, how do you are you still using the Pearson correlation?
Yes, we still use the Pearson correlation.
Okay.
Okay, thank you.
I have two questions. One is, what is the optimal size of the like this gamma? What do you think is optimal? Because if you think it's somewhere between 3 to 5 cells in one metacell, then we would have a lot of these metacells, because we generally have datasets that are in and around 100K cells. So do we deal with like 20K groups of metacells? That's first question. And second question is similar to what he has. How do you claim that genes need to be non-correlated? Because we have a lot of GRNs and we do know that some genes do control some other genes. So how will you account for those cases wherein the genes are actually being controlled by some other genes, if you think that the genes are completely non-correlated in nature?
Right. So for your second question, I think the thing you're talking about in the genes regulation relationship, where one gene regulates another gene. So those correlations those are all. I think those things are already measured in our first layer of the model, which is this expression model. So we believe all those biologically meaningful correlations or variations are already measured, are already described in this first layer of model. You see here here lambda is a p-dimensional vector. We model these p features jointly, so we already incorporated those correlations there. And could you repeat your first question? I'm sorry. Yeah.
We have a lot of cells, generally like around 100K cells. So if the metacell size is around 5, then we would have a lot of groups, right? So do we deal with like 20K groups or something like that? Or what would be a general size estimate that you would say for large scale data sets having say 100K cells, 200K cells? Like how would you say the size of the group should be?
I see. So I think for that question, that would the answer would depend on what is the aim for you using this metacell idea. If your data set is very large and your aim of using metacell is to increase the scalability, then I believe you can just choose a higher granularity level. But if your goal is mainly to solve the sparsity issue, like to find a balance between sparsity or trade-off between sparsity and the signal distortion, then I think you can still use our method to choose the optimal one to ensure that we solve the sparsity and at the same time we ensure there's not too much signal distortion.
Okay, thanks.
So the primary reason of, not primary, but like one of the reason of sparsity is also the nature of gene expression in single cell, right? There is like a lot of genes which are like have 0 and some of them have some gene expression binomial, so like Zin type of distribution. So this only take care of the sparsity in the cellular region, right? In the cell column. But what about the gene column, right?
The gene columns?
Like, yeah, here. So we still have the same number of genes. So is it possible that it's possible to sort of reduce the dimension in the gene column because you are sort of taking care of that experimental effects of correlation between the genes, right? Or is it just a cell, like you just club the cells together?
Yes, we just do aggregation on the cells dimension because here we treat cells as observations, so we believe there are only IIDs over the cells dimension, so we can only do averaging along this dimension. But I think the concept you're mentioning is something similar to the meta-gene definition, maybe. So there are also people who try to find like a gene program or to reduce the dimensionality of this genes, so that they can aggregate multiple genes into one meta-gene. I think that's similar to that concept, right?
I see. Okay, thanks.
Hi, thank you. I'm wondering, like, have you tested maybe your method on like the clonal? For example, so the ground truth that like a couple of cells, they actually have like the similar a single cell expression. So for example, like within a cell line or like a clonal of the cells, then like, and that your method can also like well capture the metacell and like not maybe clustering them into like dubious like subcluster, things like that.
Yes, actually in our paper we have an case which we analyzed the cell line data set, and we proved that we were able to we were able to identify trustworthy metacells that which is made up of single cells mostly from one cell cycle. So we can, I'm trying to say like we are able to handle multiple layers of heterogeneity. So in the cell line, we have also tried the cell line.
Thank you.
Okay, so I believe I stopped here. So this number permutation strategy is mainly designed for null construction, and our goal is to disturb those gene-gene correlation or feature-feature correlation, and at the same time preserve this the cell library sizes. So as Jessica have discussed, what we did is we first permute these genes independently within each cell, and but that would turn each column to be one perturbed permuted genes, which means that these two matrices they're not so comparable because they have different features. So that is why we need to do another layer of permutation, which is this within gene permutation on the right to serve as a normalization factor for the left ones. And this top row will form a pair, and this bottom row would form a pair. From the top row, we will get our statistics, mcDIV. And from the bottom row, we will get our null statistics, mcDIV null. So this figure is about more details of how we compute our mcDIV and mcDIV null. So we actually compute a pair of mcDIV and mcDIV null for each metacell. And here we just use two metacells for illustration, one trustworthy metacell and one dubious metacell. And this rectangle matrix represents the original data matrix with cells for rows and genes for columns. And this square matrix is the gene correlation matrix from calculated from this data matrix. So to calculate mcDIV, we use this, we calculate the Frobenius norm of the differences between this correlation matrix and the identity matrix. And we divide it by the Frobenius norm obtained from the data matrix after within gene permutation to serve as a normalization factor. And for this mcDIV null, we do the same thing, but this time, just at this time, we do it on the within cell permuted data matrix. And after we compute these mcDIV and mcDIV null for each of these metacells from metacell 1 to metacell M, we just pull all these mcDIV null values together to obtain a null distribution. And we will be able to derive a threshold from this null distribution so that we can say if a metacell's mcDIV value goes above this threshold, then it is considered as dubious. But we also noticed that this mcDIV nulls distribution is kind of dependent on the metacell size. That is why this threshold is determined as the 95th quantile conditioning on the metacell size. And by overlaying this threshold on this original statistics, mcDIV values, we can identify whether each metacell is dubious or trustworthy. So this is our method for the first goal, or say the first functionality of mcRigor, to identify those dubious metacells. And we first did a simulation using a modified version of a simulator called SCDesign 3. This SCDesign 3 was also developed by our group actually, which was published on Nature Biotechnology last year. And we modified this simulator a bit based on our two-layer model in order to generate a synthetic data set where each single cell has a ground truth metacell label. And we first noticed that our synthetic data set actually mimic this reference data set pretty well. And this somehow already shows that our two-layer model and metacell definition is somehow reasonable because they align well with the real data. And then on this synthetic data set, we first applied the general metacell methods to partition the cells into metacells. And then we applied our mcRigor to check whether each generated metacell is dubious or trustworthy. So here we first compare our mcDIV statistics values with this metacell purity, which is the highest fraction of cells in a metacell that originate from the same originate from the same ground truth metacell. Or to say this purity is like this purity kind of like represents the ground truth trustworthiness of a metacell. And we can see that these two values share pretty similar patterns, and they have a very high correlation of 0.948. So this means our mcDIV is a very good indicator for the trustworthiness of a metacell. And then using our mcRigor, we were able to classify the metacells into dubious and trustworthy with a very high F-score, like 0.921. And these gave us confidence that our method can work pretty well. And that is the simulation. We have also performed some real data validation. This one is thanks to one of our group, our lab member Manix, who is from a biology background. He pointed us to this data set with barcode multiplets. What barcode multiplet means is that in a single cell sequencing procedure, it is possible that multiple barcodes, like for example, 3 barcodes here, could be encapsulated into the same droplet with just one cell. So, and we know that in the sequencing procedure, when we later want to obtain the data matrix, we actually consider one barcode as one observation. So if this kind of technical issues happens, then we may consider these 3 barcodes as representing 3 different cells, but in fact, they just all originate from the same cell. So this is an issue, but this is good for us in the sense that since these 3 barcodes, they carry the gene expression profile from the same cell, they have the same true expression level. So these 3 barcodes could naturally be considered as be able to form like a trustworthy metacell. And this gives us a natural chance for validating our method. And it turns out that when we apply our mcRigor, it was able to flag all these 9 metacells that was formed by barcode multiplets as trustworthy metacells. So this somehow justifies that our mcRigor can reliably identify the trustworthy metacells. And another interesting application of our mcRigor is on this data set of blood cell gene expression. And these data are from both healthy and COVID-19 patients. This data was originally used to study the gene-gene co-expression differences between the 2 cohorts. Since the metacell was believed to be able to somehow strengthen the co-expression signal, here we applied the supercell method, one of the general metacell method, to this data, and we compute the gene expression over those metacells. And something in this analysis that we need to pay attention to is this gene module marked by this yellow box. So this is defined as the adaptive immune response module. So we first compare the gene expression calculated on the single cell level on the left to the gene co-expression computed at the metacell level on the right. By doing this comparison, we can see that this metacell indeed was able to strengthen the signal in both the healthy and COVID-19 groups. But the problem is that we do not really see a difference between these 2 groups in this module. So we say not enriched here. But if we apply our mcRigor method to remove those dubious metacells, we can observe, then we obtain this gene-gene co-expression in the middle. Here we can see a clear enrichment in the COVID-19 group compared to the healthy group, which implies that the genes tend to co-express more strongly in this module. And this is consistent with the fact that these genes are adaptive immune response genes. So we went back to our data to see why this is the case. And we found that the reason was that these dubious metacells, within all the metacells, like these pink one represent the dubious metacells, they falsely drove the correlations or the co-expression of genes within this healthy cohort to be very high. So these are just fake signals. And or we say there are bias in the data, and our mcRigor was able to remove them and eventually being able to identify this enrichment in this gene module. So this is like another example, but due to the time limit, I would just jump to the second functionality of our mcRigor. So far I've been talking about the first functionality of mcRigor, which is to identify dubious metacells. And we also mentioned that our mcRigor's second goal is to optimize, is to first nominate the top performing metacell method. And also at the same time, we want to optimize the hyperparameter, the granularity level for each metacell method. So the main idea here is that we will calculate an evaluation score for each metacell partition that results from a specific metacell method under a specific granularity level. And then the method and hyperparameter setting that receives the highest score would be considered the optimal setting. And now the problem becomes, how do we define this evaluation score? If you remember, when we mentioned granularity level, we said that a higher granularity level would result in bigger metacells. Which means we can better resolve the sparsity issue, but at the same time, the bigger metacells usually comes with more dubious metacells. So that is like more signal distortion. And on the opposite side, the small granularity level usually still gives more zeros, so it may leave the sparsity issue unsolved. But at the same time, small metacells may come with less dubious metacells, so less signal distortion. So that is saying like choosing an optimal hyperparameter or choosing an optimal metacell partition is more like considering a trade-off between the sparsity issue and the signal distortion. So that is why we propose two metrics, one to assess the sparsity issue, one to assess the sparsity level, which is the zero rate. That is defined as the proportion of zeros left in this metacell profile. And then we also define another metric, this dupe rate metric, which is essentially the proportion of single cells that constitute dubious metacells. So our eventual evaluation score is 1 minus the sum of these two metrics to consider a trade-off between these two metrics. And the metacell partition that receives the highest score would be considered the optimal one. And we have proved through simulation that our method is able to nominate the best metacell partition that aligns the best with the ground truth. For example, here, this ground truth gamma 50 was exactly selected as the optimal gamma by our method. Right, so to show our mcRigor's ability in optimizing the hyperparameter, let us look at an example of an important use of mcRigor, which is that we hope mcRigor could help us distinguish biological zeros from technical zeros. Here, biological zeros means that those genes are indeed not expressed at all in the cells. So these are true zeros. And for technical zeros, we mean that these genes, they are actually expressed, but just that due to the technical imperfection, those expressions are not captured and eventually shown as zeros in the observed data. So the reason why we expect mcRigor to distinguish them is that if we have like a good metacell partition where only cells of the same biological state are aggregated into one metacell, then the technical zeros will naturally go away. Because if you if a gene is expressed in the cells, in the physical cells, then just by chance, some of the cells of this state would have non-zero observed count. Then by averaging them, these zeros just naturally go away. But if it's like a biological zero, then for sure no cells of this state would have a non-zero count. So all of them should be zero, and averaging them would still give you a zero. So that is why we believe a good metacell partition should be able to preserve the biological zeros while eliminating the technological zeros. And we hope the metacells optimized by our mcRigor will be able to achieve this. So we applied our mcRigor to this data set where the where the single cell RNA sequencing data was paired with the single cell molecule fish data. The SM fish data used fluorescence hybridization to measure the gene expression. So we usually believe that these are more accurate. Usually we believe they have less, have not so many technical zeros. So here we use the SM fish data as a gold standard to quantify those zeros, whether they are biological or technical. So in our line plots here, this red horizontal line represents the proportion of zeros in the smRNA fish data, which is around 20%. So that means we believe the true proportion of biological zeros in this data should be around 20%. And then we just apply our the general metacell methods along with mcRigor. We try these three methods: metacell, C-cells, and supercell. And you can see that for each method, as the gamma value increases, the zero proportion decreases as expected. So since we mentioned that a good metacell partition should be able to preserve biological zeros while removing the technical zeros, we believe that the optimal metacell partition should be able to give a zero proportion quite similar to this gold standard from the SM fish data. And that is exactly what mcRigor told us. Here, this triangle, for example, this blue triangle represents the optimal granularity level selected for metacell, and this purple one is the optimal gamma selected for super C cells, and this is the one selected for supercell. You can see that the zero proportion corresponding to these gamma values aligns pretty well with our gold standard, this horizontal red line. So this kind of help us justify that our chosen gamma is indeed the optimal one. Well, I will also skip this example because of time limit. And so far we have been talking about the two main functionalities of our mcRigor to detect the dubious metacells and also to nominate the top performing methods and the hyperparameter. And beyond these two functionalities, a frequently asked question for our mcRigor is that how shall we deal with these dubious metacells? Because if we just naively throw them all away, then there is a big chance that we may lose some important information. Especially when we have some cells of the rare cell type, there's a high possibility that the cells of these rare cell types could be aggregated with cells of other types to form a dubious metacell. Then if we just throw it away, then there will be no information about these rare cell type left in the data at all. So that is why we propose also an extension of the mcRigor to handle these dubious metacells. And the main idea is that we try to like decompose the dubious metacells again into single cells, and then for this subset of single cells, we try to find a new metacell partitioning for them. And this partition usually consists of more trustworthy and smaller metacells. So this is like the two steps. In the first step, we first identify dubious metacells in a more conservative way by using the 85 quantile instead of 95th quantile. And then we decompose those dubious metacells into single cells again. And in step 2, we apply our selected metacell partitioning method again to this subset of single cells to identify new metacells. And we also use our mcRigor to identify the optimal granularity level. And actually, these two steps can be repeated for many times until we have 0 dubious metacells. So that is a way to handle the dubious ones. And here we apply our mcRigor 2-step on this bone marrow single cell data, which has 2 rare cell types. One is plasma blood cells and the other is HSC. And we want to see whether our mcRigor 2-step would be able to resolve these 2 rare cell types. So here in this bar plot, each bar represents 1 trustworthy metacell, and the height of the bar represents the size of a trustworthy metacell. So you can see that compared with the original partitioning, our mcRigor 2-step would be able to provide a much larger number of trustworthy metacells to work with for each of these rare cell types. So that means we have better representation for these rare cell types. And this kind of improvement is even more obvious if we look at the supercell and metacell methods. So sometimes originally we have 0 trustworthy metacells, but if we apply our 2-step extension, we have a lot more. So here comes the conclusion of our method. So far we have been able to answer our 3 questions. We have been able to give a statistical definition of metacell based on the 2-layer model, and we have detected dubious metacells using the per metacell MCDIV statistics, and we have through building the null through double permutation. And we have also been able to optimize our metacell partitioning by balancing the sparsity and dubiousness. And our paper have recently been published on this Nature Communication journal. So, and we also have quite user-friendly online R packages and tutorials on GitHub. And please feel free to check them out through these 2 links. Right. And with these, I'd like to thank our great grant sponsors, and also most importantly, thank Professor Jessica and all of our lab members from the JS lab for supporting our work. And I would also like to thank you all for your attention.